and anthers, stamens, they are all. So this is related to embryo taxonomy. And paleotaxonomy relates to pollen morphology. In pollen morphology, the units may be monad or polyad. That is also taking consideration plant taxonomy. And another one is pollen morphology. The pollen exine ornamentation and pollen aperture number, aperture position, NPC system, number, position, characters. They are all taken consideration in palynology. For example, if you take the aperture, maybe ciliate aperture, ornamentation, ciliate ornamentation, reticulate ornamentation, scabate ornamentation, echinate. So they are the different type of ornamentation. So this ornamentation and type of apertures, these aspect also the taking consideration in palynology relation to taxon. The next one, the remaining are, let's see, uh, taxon PPT. Okay, let's take the PPT slide about cytology, chemotaxonomy, numerical taxonomy. First of all, taxonomy notes, gene expression, general characters, gymnosperms. The third botany chapter, modern trends. Right. First, let's see the chemotaxonomy. So, in recent trends in taxonomy, the today topic is chemotaxonomy. First of all, what is chemo? Chemo means chemical. Chemical taxonomy. So, in chemical taxonomy, just wait. Right. Let's take pen. So first of all, So chemotaxonomy. So the chemotaxonomy, it is also called chemical taxonomy. That is the classified the plants based on the chemicals. So what are the chemicals in plants? So the chemotaxonomy can be defined as the applications of chemical data to the problems of the systematics. So first of all, what are the chemicals? What are the chemicals in plant body? How the chemicals are formed? So during photosynthesis, what are formed? Glucose formed, fructose, sucrose, they are all formed. They are also chemicals. And after during photosynthesis, proteins form. They are also chemicals. And these are the primary producers, primary metabolites. After formation primary metabolites, these primary metabolites utilized by plant body. After utilized plant body, the waste material formed in the form of secondary metabolites. Secondary metabolites. These secondary metabolites also chemical. The primary chemicals, secondary metabolites also chemical. They are formed after utilizing the primary one. The waste material, it is in the form of secondary metabolites. There may be alkaloids, flavonoids, tannins. They are all. So based on this chemical, we can classify the plants. So let's see the some of the chemicals here. Chemicals here. The here some of the potential significance and distribution of these chemicals. Chemical compounds. Already told you what are the chemical compounds. These utilized as the taxonomic data. These chemical compounds, the available for taxonomic data. Utilizing it have been utilizing since the long time. The first decondoled scientist Hoffman, Mosch are among the earlier systematic who the mainly the contribute the role for the chemistry in taxonomy. So first of all, who the mainly focusing on these chemicals, Decondol and Hoffman and Molish. These are the main scientists it work on the chemotaxonomy. Here the some of the families are given. The families are Umbelliferae. Some families mainly classified based on the chemicals Umbelliferae, Labiate, because of the fragment smell. These emit fragment smell from the crushed leaves and stem due to the presence of the essential oils. The essential oils also chemicals. The, so the based on this quick means we can identify them. So here the chemical is essential oil due to the composition of essential oil. Such families, Umbelliferae, Labiate, produces the good smell, fragment smell. The based on that, they can classify the plants. And other family in Austraceae, it has the two tribes. These are tubiflorae and liguliflorae. These are categorized based on the options of latex respectively. Presence and absence. Mainly presence. Latex also secondary metabolites. Presence and absence of latex. The presence of latex tubiflorae, absence of latex liguliflorae classified grouped in liguliflorae. So these are also chemicals. And the presence of distribution various chemical compounds. These are the already told you it is these work as a taxonomic tool, taxonomic evidence. Not all the chemical compound provide the valuable taxonomic clues. Of course, in plant body, there are several chemicals are there, but they are not used as a taxonomic aid. Only certain compounds, particularly with low molecular weight. This is important. Particularly low molecular weight, low molecular weight, low molecular weight, and which are secondary in origin and also already told how the secondary metabolites form, secondary origin of taxonomic significance. These are all. So that means the primary metabolites not used for taxonomic aid. Of course, taxonomy because almost the taxonomic chemicals almost are same in all chemicals, all plants. Sorry, all plants. But but after utilizing when the secondary production, secondary metabolites, they are the different. That's why the main consideration secondary metabolites and that secondary metabolites in low molecular weight. It should be in low molecular weight And there are total nearly 33 different groups of chemical compounds of systematic value according to Rifford et al. 1974. 1974. So this is about chemotaxonomy introduction. And next one. The few important chemical compounds are flavonoids. Just some important chemical components are flavonoids, amino acid, alkaloids, betalain, terpenoids. These are important. Let's discuss one by one. Flavonoids. Flavonoids are generally found in cell sap in vacuole. Flavonoids are developed in the chemical systematic of Asteraceae family, especially in Asteraceae family. The classification of plants mainly based on flavonoids and amino acids. Let's see here, just wait. Page number. Taxonomy. Peptide, DNA, amino acid sequence, numerical pattern. Right. Next one, amino acids. Amino acids. The presence of absence of free amino acids and their quality and quantity are of great value in chemical systematics, qualitatively, quantitatively. These both are important for the chemical chemotaxonomy. And the distribution of non-protein amino acids is very significant in the family of Leguminosae family. Non-protein, non-protein amino acid. Actually, the amino acids are protein subunit, but some amino acids do not form as a group, do not form as a protein. That non-protein amino acids a very important role to recognition of Leguminosae family. In the genus Acacia, the section Gummiferae has been unique amino acids content, and this serve as a criterion for this categorization. So there are 21 different type of amino acids are there. These different type of amino acids present different type of genus taxa. Example here section Gummiferae, one unique type of amino acids are present. And that means Gummiferae families, all plants are one type of amino acids and consisting one type of amino acid. Right, and if you take the amino acids, here the two groups of amino acids, already told you the protein amino acid, non-protein amino acid. These are the main important to classifying the plant. The protein amino acids, building units of protein, already told you, and they are released hydrolysis of protein. Hydrolysis, due to the hydrolysis, the proteins are released. Non-protein amino acids, which are not so far found in the combination in protein. They are individual and they are more numerous and estimated in about 300 in number. The occurrence of non-protein amino acids in Fabaceae male ordered Fabaceae has been attracted to the considerable interest to the taxonomist. They are present in high concentration in the seeds and amino acid content seeds in section Gummiferae of the genus Acacia is also different from that of the other section on the basis of single character alone. And that is the free amino acid lathyrine is so far known only in the genus Lathyrus. This support the present delimitation of this genus. All the chemical constituents potential useful to a taxonomic, but reality some sort of molecules are for more valuable than others. Apart from that, to organic compounds which are of relatively little use, the main varieties of compounds can be identified. They are primary metabolites and secondary metabolites and cementitides. Primary, secondary, and cementitides. The primary metabolites, the parts important metabolic pathway, and most of them are of universal occurrence and least present in wide range of plant. Example, aconitic acid, which is isolated from the aconitum. Citric acid from citrus. And secondary metabolites perform the non-vital function and are therefore less widespread in plants. Even though the rare occurrence of these among the plant, they render valuable taxonomic information. The very familiar groups of compounds have been utilized in this way include alkaloid, phenolics, amino acids, terpenoids, oil, essential oils, and carbohydrates. So this is amino acids and next alkaloids. They are widely distributed in plant families to date to nearly 5,000 alkaloids have been reported from the Angiosperm family, especially from dicots. Some families are given here, Barbadeaceae and Ranunculataceae, Leguminosae, Solanaceae are very rich in alkaloid content. The Leguminosae based on the amino acid content also classified. Amino acid content here and also alkaloid content. The alkaloid distribution has been shown to be great use in the systematic three genera of the Leguminosae, mainly Genista, Amodendrum, Adenocarpus, which have different type of alkaloids. So Leguminosae, different type of alkaloids, Genista species and Amodendron, Adenocarpus, so different types of alkaloid. So here the mainly the distribution, alkaloid distribution, the genus Homodendron, this is the Homodendron is the genus, is transferred to the tribe Genistae from the tribe Sophoraceae. From the Sophoraceae, they are grouped in Genistae because of distribution of alkaloids. Betalains, another chemicals betaline and terpenoids. First of all, there are two betalains, betacyanin. There are two betacyanin and beta-xanthin. These are the chemosystematics, which has recently emphasized by Marby These pigments are widely distributed in about 10 families. All belong to order Centrospermae. All are Centrospermae. So the Centrospermae order belong order plants mainly consisting betalains, terpenoids and steroids. Carotenoids an important group of terpenoid. Carotene, one type of terpenoid, have been proved to be the great in the family Leguminaceae Compositae. And this terpenoid also, the other secondary metabolites, one of the taxonomic value, taxonomic aid in family Garaceae, Carnaceae. On the basis of chemical evidences, Garaceae has been removed from the Hepatolus order and placed in close Cornaceae. So there the some changes have been occurred due to the composition of chemical. One group to the, which are switched to one group to the another group based on the chemical variations. So this is about chemotaxonomy. Chemical taxonomy in mainly chemical taxonomy based on chemical amino acids, alkaloids, and betalins, terpenoids, and steroids. These are all. So this chemotaxonomy also important for short answer. And here this uh, this chemistry the taxonomic distribution is not only useful the generic level but also in the family assessment. There are still considerable number of usually small angiosperm family whose affinities are not clear and which are difficult to place which certainly into the ordered system to of classification. The taxonomic relationship of Julinaceae family became a subject of controversy for least century based on wood anatomy and morphological feature associated with the wind pollination. Now this family has variously been placed in three different associations near either Anacardaceae or the Burseraceae or Jaglandaceae. And flavonoids, actually the analysis of flavonoid, carotenoid and phonoid pigment in plant of the Genseraceae have shown significant correlation with the fitting in the subfamily division of but based on geography. And another one, cementitides, actually not given here cementitides. So this is the cementitides, the human serum antibody produce antibodies as a result of introduction of foreign cell antigens. The Boyd then defined the serology as the portion of biology which is converted into the nature and interaction of antigenic material and antibodies. That means the cementide is mainly the cementide, the response of immune system, antibody. When the antibodies are produced, when antigen present, automatically antibody produced. Based on this antibody, we can classify the plants. That's also one of the taxonomic aid to classify the plant kingdom. So they are all chemotaxonomy. They are all comes under chemotaxonomy. And next one, cytotaxonomy. Another one is cytotaxonomy. Cytotaxonomy, cytological characters, which is based on wisdom. Cytological characters. What are the cytological character? The cytological character, what first of all, cytology, study of the cell components At molecular level, cell components mainly such as chromosome, number, structure, morphology, behavior. These three, number and morphology, behavior, the great help in solving many phylogenetic problems. First, let's see chromosome number. The, the importance of chromosome number to taxonomy has been known for more than century. In the flowering plant, the lowest chromosome number is found in Haplopappus gracilis, which is lowest 14, while the highest chromosome number found in Poelytrosa, 265. Many of the recent published generally include the chromosome number of species listed. The chromosome number may or may not vary within group. It is not taking important consideration, because if you take the number of family, then they not be unique. Sometime in the members of the Fragaceae family, the chromosome number may be remain constant, 12. In such cases, chromosome number is of little help for it. Cannot be distinguished the taxa. That means all number of chromosome 12 diploid state in one group. The genus Salix and basic chromosome number is 19. But the species from the polyploid series, for example, S. viminalis, diploid 38 chromosome number, Arctocinera, diploid 76 chromosome number. So this is the variation in chromosome number. Chromosome number may be, these are the dynamic within the group of the family. Next. And the polygonum of amphibia, there are the three varieties. These some of the morphological different when grow under different climatic condition. On cytological analysis, these were the found to have identical chromosome number. Now chromosome morphology. Chromosome morphology, one of the important consideration is providing taxonomic evidence, size, length, from and amount of distribution, heterochromatin, all the criteria. So size of the chromosome important, length of the chromosome important, and the amount of distribution of heterochromatin. If you take the chromosome, there are two type of chromatin, heterochromatin and euchromatin. So the also the heterochromatin also utilized for the taxonomic classification, taxonomic plant classification. First, let's see the size different within the group, length also different, and distribution of heterochromatin. That means distribution of heterochromatin, well, apart from the euchromatin. What is the euchromatin, heterochromatin? Euchromatin, if you take for example, where is well illustrated. Right. Size also considers various in size and also length, size and length, the amount of distribution of heterochromatin. They are the criteria. Heterochrom, for example, if you take the uh, chromatin, this chromatin, in this somewhere dark color region there and light color, and again dark color, light color, dark color. So the distribution also, the dark and light color distribution, this dark color heterochromatin, light color euchromatin, and this dark and light color combination also utilized for the taxonomic classification. The mostly the monocot usually have the longer chromosome than dicot. So this is also one of the consideration. If, for example, if you find out the new plant, if the chromosome number long, that is grouped in monocots, small in size, they are mainly present in dicots. Many families, like a given Ranunculaceae family, chromosome types may give and indicate the evolutionary trend. The presence of absence of B chromosomes, mainly the B chromosome present in plants only. B chromosome presence and then frequency in particular population provide useful taxonomic clues. And chromosome behavior. This is the chromosome morphology. What you mean? The mainly chromosome morphology, size-wise, length-wise, and distribution of number of heterochromatin. For example, take the size of the cell, size of the chromosome, almost. If you take only, of course, in variations in dicot and monocot also, mainly if we broadly classified the dicot, monocot have the longest long chromosome than the dicot, right? And B chromosome presence of B chromosome is also one of the criteria. And chromosome behavior. The behavior of the chromosome during meiotic phase, especially during the pairing, will indicate the hybrid characters of the individual. The chromosome behavior, that means the pairing occurs in zygote stage, that is called synapsis process. And pairing also, the formation of pairing of chromosomes also shows the variation in one family to another family, one species to another species. So that is also taken consideration. And cytological variation of the family, the arrangement of the plants within family according to their phylogenetic well, according to the phylogenetic relationship has been greatly helped in cytological evidence. The arrangement of genera tribes in the family Cruciferae, Graminae, and Ranunculaceae have been based on cytological evidences. Cruciferae arrangement, Graminae arrangement, and Ranunculaceae, they are all based on phylogeny. The phylogeny based on chromosome size, structure, the cytological variation at general level. Here the given some of the general levels. The Cruciferae 2 Physaria and Lisquerella were identified as a single genus by many botanists. Cytological analysis has supported separation of the two in two different genera. Cytological cultivation specific level. Next numerical taxonomy. Next I will cytological variation at specific level. There is a great amount of cytological variation according to the specific and intraspecific level, not only specific, but also intraspecific level. The different species of the genus Narcosus of the family of Amaryllidaceae have been arranged according to the phylogenetic relationship on the basis of karyological study, nucleus. Cytological data are made to use in the system of classification of the Cronquist. Cross classification. In cross classification, mainly taking consideration cytological data and numerical taxonomy. There some another take another one. I will numerical taxonomy. Another slide is good. Chapter 6 modern taxonomy. Where is Betham, Lambda Monas, Teenager, Albugo, enzyme used GE, Albugo, cell biology, nomenclature, modern trends in plant taxonomy. So numerical taxonomy, another name phenotics. Pheno to appear. Also another name taximetric. It attempt to classify the organism based on the overall similarity. This is important. Based on similarity. Based on and another name. Remember this one also. Don't get confusing with these words. Phonetics or numerical taxonomy. Both are same. And also called as taximetric. Taximetric. And it is an attempt to classify the organism based on overall similarity. Similarity, morphology, observable traits, regardless of their phylogenetic and evaluation relations. And already told you the phenotics are also called numerical taxonomy. As against the phenotypic system, the phylogenetic system, the emphasis on the evolution relations among the organism. So in phenotypics, in numerical and phylogenetic system, mainly the focus on evolution relation among the organism. Biometric or taximetric is the statistical research on biological phenomena. It is the statistical research, that's why it is called numerical taxonomy. And numerical taxonomy is the technique of classifying the plant using numerical method. The present it is also called phonetics or taximetrics. Phonetics is the numerical evolution of the similarities or affinities of taxonomic units, which are typically classified into taxa based on their affinities. If the affinities means the descendants, common descendants group of the plants taxa, that is called affinities. For example, here the one plant. One, two, three. Again these. So again some other. So these relations called as affinities from one common ancestor. So these are the from common ancestor. These characters carry forward from the common ancestor. So this is called affinities, which are typically classified into taxa based on their affinities. These affinities classified into taxa. They are concept. First of all, this concept was proposed by Michael Adamson. Michael Adamson, a French botanist, 18th century. Therefore, this is called as Adamsonian taxonomy. Another name, Adamsonian taxonomy. And Smith and Shokal published a paper, Principles of Numerical Taxonomy, in 1963. Peter Smith and Robertson. Some of the important terms here: phenotic relationships, phylogenetic relationship, cladogram, clade. The first of all, phenotic relationship. The relationship. Just will take a pen. The relationship among the organism based on the morphological and observable traits. That is phenotic relationship. Phenology, study of morphological study. And phylogenetic relationship, the relationship among the organism based on the degree of relatedness, degree of relation due to the common ancestor. Already draw this one, ancestor, two, maybe four, maybe five. So the relatedness, degree of comparison, relatedness of degree of characters from common ancestor A, B, C, next D, E, F. So the relatedness also. And cladogram. Cladogram means evolutionary classification. Evolutionary classification generate trees and called cladogram. Here the cladogram evolutionary classification of generates trees and called the cladogram. Clade is group of organism. Clade is group of organism. Clade group of organism that includes an ancestor and all its descendant. That's it. And ancestor and all descendant like this. This is clade. Next one. They are not important. Basic common definition, scientific study, data, numerical, numerical phenomena, descent, evolution, and next numerical taxonomy. Now in brief, in this type of classification based on analysis, let's take unless taxonomic data, this numerical comparison takes place of equally weighted character by mathematical and computerized method. In this evaluation, mainly mathematical, not only mathematical and computerized method. This numerical evaluation, the similarities or affinities between taxonomic units, they are arranged in the taxa at the base of their affinities. This concept was proposed by Adamson and later Smith and Sokal published a book, Principal Numerical Taxonomy. I already told you. I repeated this one. Principles. The more character, the better given classification will be. Every character should be given an equal weightage. The classification based on the phenotypic similarities. Phenotypic similarity means morphological similarities. Next one. And for numerical taxonomy, some of the important criteria or aspects are important, such as operational taxonomic unit and selection of operational taxonomic, selection of characters and traits, cluster analysis by computer software, determination taxonomic relations between OTU, that means uh, next classification based on the base of overall similarities. Now selection of characters, before selection of characters, selection of taxa, OTU, that is operational taxonomic unit. What is operational taxonomic unit? That is simple. Any sample or item or individual which is selected used for analysis is called as OTU. That's the first taxonomic aid. Any sample, any plant sample, any item, what is going to be classified, that is OTU. And the next, it is the basic unit of numerical taxonomy, because based on this, we can classify the plants. Next one. It may be individual or species, genus, family, order, or class. So that means if you take one plant taxa, that is also OTU. Genus, OTU, and family member, complete family, order, they are also OTU. Anyone which are you are utilized for the taxonomic classification. Anyone that is utilized for taxonomic classification, that is called OTU. In case OTU is above level individual, then adequate relation to various form, because when, for example, genera is OTU, then it should be represented by different species. Here the selected one genera, for example, uh, one of the genera, like one Vahinia, for example, same species Genesis alpina, Pulcherima, right. Senna, Senna arculata, Senna fistula. Senna is the genus. Actually, the Senna genus is the act as a OTU, and different species. Arculata. Next one, Senna fistula, and another one. Yeah, another one. Just wait. Senna species. Senna is the genus, that is clear. Senna species that are given here. Senna auriculata, Senna alata, and Senna roxburghi. So more than in Senna, almost 200 above different species are there. So, but genus is the Senna, that is act as a OTU. Methods and procedures. Numerical taxonomy. First, already told you selection of taxa. OTU. The taxa may be genus level or species level or family. Next, selection of characters or traits. After selection OTU. Next, selection of characters. Cluster analysis via computer software. Determination of taxonomy relation to between OTU. Classification based on the basic overall similarities. First one, selection of characters. If the characters which are used for called unit character, these unit character classified into binary character or qualitative multi-state character and quantitative multi-state character. First of all, what is binary character? A binary by means namely given to a character only having two states and recorded at present or absent. That means only two states, whether it is present or absent. That is binary character. Present which represent with plus or one, Absent represent with minus or zero. Missing, that means that character not appeared, that is no comparison. NC, no comparison. That is the one of the method. Selection character. Another selector character, qualitative multi-state character. Multi-state here. Here binary, bi-state. Here the multi-state. And here is also multi-state. But one is the qualitatively, another one is quantitatively. First, let's see the qualitative. Qualitative. This character possesses three or more contrasting form. Here the multi means more than two. The more than two characters here given the flower color red, white, yellow. Color one character, but it is based on different type of color. Red color, white color, yellow color. That is the quality. Now it is quantity number. The number which is represents the represent the measures of the size of continuous scale, weight or length. Example, the length of the spore, amount of chemical produced by bacterial strain. These are the quantitative. This is quantitative. Quantity, length, quantity, short or big. Which amount produced by bacterial strain. That is also quantity. So these are the quantitative multi-state. Now the rules for selection unit character for numerical taxonomy. But the rules are must include all parts of the organism and not less than 50 character. Here the character not based on one or two character. 50, 50 minimum. And also you should take in consideration all part of the organism. If you take the, for example, root, plant, root, take in consideration. Stem, leaf, flower. And also in molecular level also. Chemicals also. They are all taking consideration. And also the comparison character minimum 50 character, 50 minimum. And belongs to all stages of life cycle. It is not only confined to vegetative stage, not only for reproductive stage, all stages. So due to this, that means take consideration morphology, physiology, ecology, distribution, all. And character weightage, how can give weightage? The two type of weightage, equal weightage and successive weightage. In equal weightage means all characters selected are given equal weightage while creating taxonomy groups and is one of the principal number of taxonomy. First of all characters selected. In successive weightage, the character which shown the least unique and identified and given more weightage is the subsequent analysis. In equal all character, here the least character, that is the importance. Now the data processing, here the one side given characters, here taken 10 characters. And this is taxonomic units, here the taxonomic units A, B, C, D. First one character. This one character present in A, OTU, B present, but C absent, 0. And D, new character, that is different one. No correlations. LC, what is given here? No comparison. No comparison. Present or absent, that is okay. But no present, but it is different character. That is, we can't compare the remaining one. For example, this is one of the character. For example, so for example, cartiladons one. For example, one cartiladon. This is also one cartiladon. But this character, no cartiladons, not present. 0, cartiladon character 0. But here the number of cartiladons different, all the more than 2 cartiladons. So there is no comparison. If we take the number of cut letters, no comparison remaining A and B and C. And second character present in A taxonomic aid, B taxonomic aid, C and D, all consisting all. And third character same, 1 and 2 and 3. Fourth character first absent, only B present, no comparison in C and D. So this is the classification. Already given NC for no comparison, 1 is present, 0 is absent. Estimation similarity, similarities, and again taken 1, 2, 3, 4, 5. Similarities minus no similarity, no characters. So here the similarities only 2 present, 3 present, 2 and 3 same character present. And here the one character absent, one character present. Here 2 character absent. So the OTU scored for the 5 characters. These are agreed second and third here. Second and third. Out of 5 only third and also second and third and fourth and disagree in first and fifth. If you take first and fifth here. So actually these two all character present in, sorry. Two and three all character present. Second and third. Fourth, first, first and fifth. Actually, if you take fourth, no, disagree. Fourth, disagree. If you take first and fifth, here present, one absent. So similarity, total 5 characters. In 5 characters, similarity character 3. 3 similarity characters. So total person 60. And dissimilarity characters 40. Dissimilarity character 40. Dissimilarity here, dissimilarity 1. These two also 1. Total 2. 2 by 5, 40%. So this is the estimation of similarities. This qualification not important. Taxonomic structure. The final one, taxonomic structure. The similarities matrix, resemblance matrix is the basic evidence of available for finding taxonomic structure. Cluster analysis, it is also not important. And now let's see the advantages of numerical taxonomy. Provide results that agree with established classification. Highly relevant to real problem biological classification. Provide a more exact basis for classification. Integrate data from various sources from morphology, physiology, biochemistry, DNA. Already told you we can include in taxonomic, include all plant parts, and also take in consideration all care more than 50 characters. B, quantitative the methods provide greater discrimination along the spectrum taxonomic differences and re-examinations of principles of taxonomy. These are the advantages. We can re-examine because it is of the data present, taxonomic data present. So we can re-examine the entire taxonomic classification. Now let's see the demerits. It is useful phonetic classification, but not phylogenetic classification. Numerically okay, but where is phylogenetic relation? Where is phylogenetic relations in affinities? Character selection is crucial because the character chosen for comparison are inadequate. The statistical method may give less satisfactory solution. Different taxonometric producers may give a different result. That is also important. So the numerical, some of the demerits, not for phylogeny, here the crucial important character because it is observation and similarities, morphological and observations. Different taxonometric procedure may give a different result. For what, for example, one family, different one within the family, number of plants are there. For example, you take the chromosome number. The chromosome may be variation within the group of the plants. So that is the demerits. Next, right. And and apomorphy, geomorphy is not important. What is clad? Already one second discussed phenogram and cladogram. Phylogenetic tree. So phylogenetic tree, which means simple phylogenetic tree and a visual represent of the relation between the different organisms, showing path through evolution time from common ancestor. Here the root node. So here the given common ancestor of A and B and C. This is the root node. One common ancestor A and B. And this is also common ancestor to C. C is the outgroup to AB. But actually, this is one same group, AB same group, C outgroup, but ABC common root node. ABC common root. That is the phylogenetic tree. And these are not important in theory exam, but remember for competitive level. Phenograms and cladogram. Phenogram. It is type of dendrogram which is based on phenotypic data. That means morphological and observation. The lines called the phenon lines, drawn at right angles to the dichotomously branching dendron represent the percentage similarities. Already told you similarities of phenotypic features between the organisms. Cladogram, it is the dendrogram that relay an assumption about the ancestral relationship, one genre to another genre, as well as current data, from ancestor to current data. The phenotypic data are based on features of organism, whether they are analogous or homologous. What are analogous character? What is homologous character? Before analogous and homologous characters, here dendrogram. So what is the dendrogram? Dendrogram, that is the dendrogram simple diagram that shows the hierarchical relation between the object. That is called dendrogram. So in phenogram, this graphic representation based on phenotypic data. That means phenotypic data means the morphological observation data. But in cladogram assumption, if the clear data, phenogram, clear data based on clear observation, we classified the plant. But in cladogram, some of the assumption there. So maybe you don't know ancestor plant, but you know the present data. Based on current data, you can draw, you can assume the ancestors, affinities of that plant. and phonetic data based on features in organism regardless whether they are analogous or homologous. So here the difference between analogous and homologous. For example, best example analogous, homologous. Um, if any two structures or organ perform similar function similar, but are structural different, that is called analogous. In analogous, uh, analogy, homology is given importance. If you know data, features, organism, regardless, analogous, homologous. Here analogy means function, F for function, structure, A for analogous. In analogous, similar function, body part, here the part. For example, here the A and B, two organisms. Function same function. Structure different. Structure different. Function F for same function, A, B, same function, A part, B part, function same. But structure different. That is analogous. In homologous, structural similar, reverse. Structure similar, function different. Homologous. Homologous means homologous, structure similar, function difference. Analogous, function same, structure different. For example, best example, let me give from the uh, example wings of bird. Take the animals, wings of birds and insect. Actually, wings of birds, so insect wing. These are analogous or homologous. Actually, these wings of bird, insect wings, function same, but structural different. That is, they are analogous. And the homologous example, homologous means structural similarity, function different. Mm, For example, homologous, homologous structure same. Maybe one example. Wing flying animals like birds, insect. Okay. So if you take in plant kingdom, plants, rhizoids, rhizoids and roots. Root structure different, rhizoid structure also different, but function same. Function same. So they also analogous. And homologous example. So two example for analogous completed and homologous example in biology. Anaptically same structure but different function called homologous. Example, uh, mouth parts of cockroach, honeybee. So mouth parts of cockroach and mouth parts of honeybee, butterfly, forelimb of man, bat. Thrones of in botany, thorns of bougainvillea, tendrils of cucurbita. Actually, these are homologous. For example, thorns in, let me write, anatomical structure thorns, bougainvillea, tendril, cucurbita. This anatomical structure same, but function of thorns that is for protection. Function of tendril for climbing. Function different. The homologous function different. Function different. But if you take the structure, the thorns and tendril, the development, anatomically, they are the similar structure. Maybe some variation in external, but anatomically similar structure. That is homologous, but the function different. Thorn function, that is not for climbing. Tendril function, not for protection. That's it. And if you take the in animal, mouth parts of cockroach, honeybee. So mouth parts cockroach same, honeybee internal same, forelimb of man same, but their functional all different. Functional variations are there. So that means in phenogram, there is no regard, there is no consideration analogous and homologous character. But in cladogram, the analogy, homology given importance, and organisms that are evolutionarily distant may be grouped closely together. It is not so that here given phenogram example, gymnosperm tree evolved in Carboniferous period, whereas angiosperm tree did not evolve until 90. But in phenogram, they will be grouped together on the basis of structure. OTU, operational taxonomy already told you. OEU, operational evolutionary units. And what is clade? Already told you. Clade, not clade here. The group of species that include single common ancestor and are all descendant of that ancestor. That is called clade. For example, you can see here single ancestor to here. To one to here. So these are linked to here. This also one ancestor, one same to here, one to can see here different. Here also different. No relations here and here. So they are the not clades. Isomorphy, apomorphy refers to novel evolution. Apomorphy, novel evolutionary characters. They are novel, new evolution character unique to particular clade. This novel unique character, particular clade. It is it and it all its descendant. Presence of feather in aves, absence of legs in snakes. So that means refers novel new characters, new evolution character, unique particular clade, and all its descendant. In restricted single species called automorphy, epomorphy, one single class automorphy, presence of feathers in aves, absence of legs in snake. They are the new characters. All aves should consist in feathers. That is also new evolution character. But these legs are not present in snakes. That is also one unique character, new character. Plesiomorphy. It refers to evolution character homologous within the particular clade. I already told you what is homologous. Structural difference. Structural same, function different. And within a particular clade, but not unique. It's not unique to all members of that particular clade. Presence of legs in reptile. Is it unique character? Take reptile, snakes no legs, but in lizard it have the leg, crocodile leg. So this is the plesiomorph. Different group of organism: monophyletic, paraphyletic, polyphyletic. Monophyletic group includes and one ancestor here, mono, and all its descendant. One, A, B, C, D. They are all one ancestor. Paraphyletic includes ancestor and some, but not all of its descendant. But they are all not a descendant. So that is paraphyletic. Polyphyletic means more than two different groups. More than two different groups. Here the A, B one group. So A and C one group, B and D different. So that different one. That is called polyphyletic relationship. Next one. Screen document advanced. Open background. Where is screen? PPT whiteboard. Explain it. General accounts of Ranels, Centrospermae, and Amentiferae. First, let's see the Ranels characters. Let's change color. Ranels characters. So mainly based on in Ranels characters are number one, numerous infinity floral characters. That first one. Second, floral parts arranged in spiral, hemicyclic, hemicyclic manner. And third character, flowers are hypogynous. This is the hypogynous symbol. And with much elongated convex, perianth generally petalide and perianth, petalide perianth. Generally petalide, perianth generally petalide, maybe or perianth difference into calyx or corolla. And pollination by beetle, Cantherophily, Cantherophilus, Cantherophilus. It mainly this Ranels consisting eight families such as Magnoliaceae, Anonaceae, Dillinaceae, Calanthaceae, Gymnospermaceae, Nymphaceae, Ranunculaceae, Barbadeaceae. The earlier family of the order include woody plant with oil-containing cells in the parenchymatous tissue. In the latter families, oil-containing passage are absent, and the plants are mostly herbaceous. So first of all, let's key, indented key, first character, first character, first A, trees, essential oils in parenchyma, in parenchyma. And this one, first A, two, maybe A or B, sepal three, and another two, sepal five. So this is number three. Go to three, sepal five in this. Then 2A, 2B. Actually, go to 3 is 2A, 2B. Go to 3, sorry, this one, go to 3. So 3A, 3B. Sepal 5, go to 4. Order number 4. This 4A, 4B. Always 2 characters. In 3A, stipule present. Sepal 3, stipule present. Endosperm, not ruminate. So that is Magnoliaceae family, Magnoliaceae. Same sepals 3 here also. Stipules absent. Stipule absent. But endosperm ruminate. That is Anonaceae family. Sepals 5. Numerous. Go to 4. Next order. 1. Next to 2. 2 go to 3. 3A. 2A. Here also same 2A. 2B. In 2B. Go to 4. 4A. 4B. In 4A. Sepals 5. Sepals numerous. So here the character sepal 5 to numerous actually. 5 to numerous. Sepal 5 Diliaceae family. Sepal numerous Caliocanthaceae family. So like this we can classify the indented key to all 8 families. Magnoliaceae and remaining families also we will discuss tomorrow.